Hello everyone, this is Deepak sir, biofaculty for NEET exam and CBSE board exam. Now I'm going to discussing about PYQ of board exam for chapter molecular basis of inheritance. तो इस वीडियो को देखने के बाद यानी अगले 1 घंटे बाद ये आपका चैप्टर के अंदर जो डर है एग्ज़ाम को लेकर के वो पूरी तरह से ख़त्म हो जाएगा. So let's go started. First question is how many base pairs would a DNA segment of length 1.36 mm have? Here length of DNA is given, and in these questions we apply the formula of length of DNA segment equals number of base pair into distance between two base pairs. After applying this formula you get the answer. Very simple. Second questions is the same on the basis of this formula. In a experiment, DNA is treated with compound which tends to place itself amongst the stock of nitrogenous base pairs, as a result of which the distance between two consecutive base increases from 0.34 nanometer to 0.44 nanometer. Calculate the length of DNA double helix which has 2 into 10 to power 9 base pair in the presence of saturating amount of this compound. So after applying same formula you can get the answer. Next question, third, calculate the length of DNA of bacteriophage lambda that has 48502 base pairs. So here number of base pairs is given. So this is the same uh, formula based questions. Now next question number 4. Why are proteins either positively or negatively charged? So answer is the if the proteins are rich in basic amino acids, they are positively charged, and if the proteins are rich in acidic amino acids, they are negatively charged. Now next question 5. Mention the carbon position to which the nitrogen base and the phosphate molecule are respectively linked in the nucleotide given here. So you can see in this diagram a sugar connected with base at the carbon number 1 and carbon number 5 at a carbon number 5 phosphate group is linked. Now question number 6 is the same question. What is the A and B? You see in this diagram the B is nitrogen base and A is phosphate group. Next question number 7. Name the negatively charged and positively charged components of nucleosome. So very simple questions. Negatively charged is DNA and positively charged is histone octamer. Now next question is 8. In a molecule the number of RNA nucleoside triphosphate is 10 times more than the number of DNA nucleoside triphosphates. Still only DNA nucleotides are added during the DNA replication and not the RNA nucleotides. Why? So answer is DNA polymerase is highly specific to recognize only deoxyribonucleoside triphosphatase. Therefore, it cannot hold RNA nucleotides. Next question number 9. Name of the transcriptionally active region of chromatin in a nucleus. So answer is euchromatin. Now next question 10. Mention the two additional processing which hnRNA needs to undergo after splicing so as to become functional. So answer is capping and tailing. Now next question 11. When at when and at what in does the tailing of hnRNA takes place? The answer is tailing of hnRNA takes place during conversion of hnRNA into functional mRNA. After transcription, it takes place at 3' end. Now, question number 12. At which end do capping and tailing of hnRNA occurs respectively? The answer is capping occurs at 5' end and tailing occurs at 3' end. Next question number 13. What is cistron? A cistron is a segment of DNA coding for polypeptide. Next question number 14. How does a degenerate code differ from an unambiguous one? Answer is degenerate code means that one amino acids can be coded by more than one codon. Unambiguous code means that one codon codes for only one amino acid. Okay. So when one amino acid coded by more than one codon, then called degenerate. Okay. And then and when one codons code only one amino acid, then known as unambiguous. Okay. Now next question number 15. Mention two functions of the codon AUG. The answer is two functions of the codon AUG are: first, it acts as a start codon during protein synthesis. Second, it codes for the amino acid methionine. Next question, number 16. Write the function of RNA polymerase second. RNA polymerase second transcribed precursor of mRNA or snRNA. Next question number 17. Give an example of a codon having double function. The answer is AUG. AUG acts as initiation codon and also codes for methionine. So AUG have double function. Next question number 18. Mention how does DNA polymorphism arises in a population. DNA polymorphism in a population arises due to presence of inheritable mutation at high frequency. Now, next question number 19. Suggest a technique to a researcher who needs to separate fragments to DNA, fragments of DNA. The answer is gel electrophoresis is used to separate DNA fragments. Next question number 20. Mention the difference to distinguish an exon from an intron. The answer is exon is the coded or expressed sequence of nucleotides in mRNA. Introns is the intervening sequence of the nucleotides not appearing in processed mRNA. RNA. Okay. Now, question number 21. What do X and Y represent in the transcription unit of DNA molecule shown? So you see here in this two box given in the diagram, here A is mentioned and X did mention and Y. So X is template strand, X is template strand, Y is terminated. Okay. Now, question number 22. Why HNRNA is required to undergo splicing? HNRNA undergoes splicing in order to remove introns. Okay. HNRNA undergoes splicing in order to remove introns which are intervening or non-coding sequences, and exons are joined to form functional mRNA. Now next question 23. Given below is a schematic representation of a lac operon in the absence of a inducer. Identify A and B in it. So you see here, this is lac operon and this Pipu Jaya. So in this diagram, what is A and B? So A is repressor and B is repressor bound to the operator and prevents transcription of structural genes. Now, question number 24. Why is lactose considered an inducer in lac operon? So, lactose binds to repressor and prevents its form binding with the operator. As a result, RNA polymerase binds to promoter operator region to transcribe the structural genes. Next, question number 25. State which human chromosome has A. The maximum number of genes and B. The one of which has the least number of genes. So A chromosome first and B Y chromosome. Next question number 26. How is how is repetitive or satellite DNA separated from bulk genomic DNA for various genetic experiment? So answer is density gradient centrifugation. Question number 26. Okay, now next question number 27. Mention the contribution of genetic maps in human genome project. So genetic map have played an important role in the sequence of genes, DNA fingerprinting, tracing human history, chromosomal location for disease associated with sequence. So now I'm going to discussing short answer. Okay. So these questions gaining 2 marks. Okay. So first question is draw the schematic representation of dinucleotide level the following. A. The compound of nucleoside. B. 5' end. C. N glycosidic linkage. And D. Phosphodiester linkage. So this is a very simple question. we know that what is nucleotide? Nucleotide equals ribose sugar plus base plus phosphate group. You see here, this is sugar, pentose sugar. Pentose sugar have 5 carbon. First carbon linked with the nitrogen base and 5 carbon linked with phosphate group. And this nucleotide linked with other another nucleotide to form a long chain called polynucleotide, or also you know about nucleic acid. So very simple. Now next question is second, describe the structure of RNA polynucleotide chain having four different types of nucleotides. So this is the same pattern question. You see here many nucleotides linked with each other to form nucleic acid. So the first nucleotide linked with other nucleotide by 3 to 5. Okay, and if first nucleotide linked with other at carbon number third to carbon number 5. So in this type of pattern, repetitive. So 3 dash to 5 dash and 5 dash to 3 dash. So very simple. And next question, third. This questions asked CBSE Delhi 2015. A DNA segment has a total of 1500 nucleotides, out of which 410 are guanine containing nucleotides. How many pyrimidine bases this DNA segment possesses? So here we apply Chargaff's rule. So according to Chargaff's rule, adenine by guanine equals thymine by guanine equals 1. So you see here G equals C and G equals 410, hence C is also equal to 410. So G plus C equals 410 plus 410 equals 820. So A plus T equals 1500 minus 820 equals 680. Now as we know A equals T, so T equals 680 by 2 equals 340. So pyrimidines equals C plus T, 410 plus 340 equals 750. Now coming to next question, number 4. A DNA segment has a total of 2000 nucleotides, out of which 520 are adenine containing nucleotides. How many purine bases this DNA segment possesses? So this question asked CBSE Delhi 2015. Here same formula will apply like above the question. A equals T, A plus T equals 520 plus 520 equals 1040. Total number of nucleotides 2000. So G plus C equals 2000 minus 1040. Now obtain 960. G equals 960 by 2 equals 480. Total number of purines A plus G equals 520 plus 480. So the finally get result 1000. Now next question, describe the structure of nucleosome. This refers to number 4 number. Okay. Next question number 6, draw a schematic diagram, a part of double-stranded dinucleotide DNA chain having all the 4 nitrogenous base and showing the correct purity. So same questions asked CBSE Delhi 2012. So this is the same question of refer to figure 5.3. Now question number 7. Although a prokaryotic cell has no defined nucleus, yet DNA is not scattered throughout the cell. Explain. DNA is negatively charged and has positively charged non-histone protein. This helps DNA in be held in a place in large loop called nucleoid. Now next question number 8. Explain the role of sulfur 35 and phosphorus 32 in the experiments conducted by Hershey and Chase. The answer is viruses grow in the medium containing phosphorus 32 containing radioactive DNA but not radioactive protein, because DNA contains phosphorus but proteins do not contain phosphorus. So similarly, virus grow on radioactive sulfur containing radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Now next question number 9. Recall the experiment done by Patrick Griffiths, Abdi, McLeod and M. Carty, where DNA was speculated to be the genetic material if RNA instead of DNA was the genetic material. Would the heat-killed strain of Streptococcus have transformed the R strain into virulent strain? Explain the answer. So here RNA is more labile and prone to degradation owing to the presence of 2-hydroxyl group in its ribose. Hence, heat-killed S strain may not have retained its ability to transform the R strain. So now coming to the next question number 10. How do histone acquires positive charge? Very simple question. The answer is histones are rich in basic amino acids. Histones are rich in basic amino acids, residues, lysine and arginines, which carry positive charges in their side chains. Therefore, histones are positively charged. Now next question number 11. A single base mutation in a gene may not always result in loss or gain of function. Do you think the statement is correct? Defend your answer. So answer is, the statement is correct because of degeneracy of codons mutations at third base of codon usually does not result into any charge in phenotype. This is called silent mutation, but at other times it can lead to loss or formation of malformed protein charging the phenotype. Okay. Now next question is 12. Protein synthesis machinery revolves around RNA, but in the course of evolution it was replaced by DNA. Justify. Answer is, since RNA was unstable and prone to mutation, DNA evolved from RNA with chemical modification that makes it more stable. DNA has double-stranded nature and has complementary strands. This further resists charges by evolving a process of repair. Next question number 30. Explain the two factors responsible for conferring stability to double helix structure of DNA. Factors responsible for conferring stability to double helix structure are presence of hydrogen bonds, the plane of one base pair stacks over the other, complementary presence of thymine in place of uracil. Next question number 14. Here A dash DNA dash mRNA dash protein. So look at the above co-sequence, mention the events ABC. What does central dogma state in molecular biology? How does it different in some viruses? So answer is A DNA replication, B transcription, or C translation. Central dogma in molecular biology states that information flows in the order DNA to RNA and RNA to proteins. It differs in some viruses as the flow of information is in reverse direction, that is, it's from RNA to DNA. Next question 15. Retrovirus do not follow central dogma comment. Genetic material of retrovirus is RNA. At the time of synthesis of protein, RNA is reverse transcribed to its complementary DNA, first then transcribed to RNA and proteins. Hence, retrovirus are not known to follow central dogma. Okay, very simple. Next question number 16. Here the replication fork structure is given. Why do see two different types of replicating strands in the given DNA replication fork? Explain, name these strands. So these questions asked CBSE Delhi 2011. So answer is, the DNA dependent DNA polymerase catalyzes polymerization only in one direction, that is 5' to 3'. Therefore, in one strand with polarity 3' to 5', continuous replication takes place, whereas the other strand with polarity 5' to 3' carries out discontinuous replication. The strand with polarity 3' to 5' is called leading strand, and the strand with polarity 5' to 3' is called lagging strand. Now, question number 17. Name indicating their functions a few additional enzymes other than DNA polymerase and ligase that are involved in the replication of DNA with high degree of processability and accuracy. So here answer is first helicase. Helicase helps in opens the helix. Second, topoisomerase removes the supercoiling of DNA, relieves the tension due to unwinding. Third is polymerase, which helps in synthesis of RNA primer. Fourth, telomerase to synthesizes the DNA of telomeric end of chromosomes. Now, next question number 18. During in vitro synthesis of DNA, a researcher used 2-3-deoxycytidine triphosphate as raw nucleotide in place of 2-deoxycytidine triphosphate. Other conditions remaining as standard will further polymerization of DNA continue up to the end or not? Explain. So answer is further polymerization up to the end would not occur as the 3-OH on sugar molecule is not for forming ester bond, which is required to add another nucleotide. Next question number 19. State the double role of deoxyribonucleotide nucleoside triphosphate during DNA replication, or write the double purpose served by deoxyribonucleoside triphosphate in polymerization. These questions asked CBSE in 2018. The answer is deoxyribonucleoside triphosphates act as substrate for polymerization, and second, these provide energy from its two terminal phosphates for polymerization reaction. Now coming to the question number 20. Discuss the role of enzyme DNA ligase plays during DNA replication. The answer is DNA ligase joins or seals the discontinuous DNA fragments. Next question 21. Given below is one of the strand of a DNA segment. So here 3 to 5 is given, and question A, write its complementary strand. Question B, write a possible RNA strand that can be transcribed from the above DNA molecule formed. The first answer is 5 to 3 the sequence of bases ATGC, ATCGC, TGC, TGC. Purity half, nucleotide sequence half. B 5 to 3 AUG, CAU, GCAU, GCAU, GC. Polarity half, nucleotide sequence half. So this question asked CBSE exam 2020. Now next question number 22. The base sequences of one strand of DNA is TACTAZGAT. Write the base sequence of RNA got after transcription of the given sequence. Next question B is what is the distance maintained between the two consecutive pairs of bases in the DNA molecule? And C question who contributed the base complementary rule? So answer A is AUG AUCCUA. B answer is 3.4 angstrom or 0.34 nanometer. Option C is Chargaff contributed the base complementary rule. Now next question number 23. Draw a level schematic diagram of transcription unit. So the refer to figure 5.10. Next question 24. Explain the process of transcription in a bacterium. The answer of this question refer to point remember 13. Now next question 25. In this question there are two questions given. First A is construct a complete transcription unit which promoted and terminated on the basis of the hypothetical template strand given below. So you see here ATGC ATGC ATAC and the question B write the RNA strand transcribed from the above transcription unit along with its polarity. This question asked CBSE Delhi exam 2012. So here you see answer is A, answer of 3 dash to 5 dash ATGC ATGC ATAC and promoted and terminated is shown in the figure and answer B is transcribed RNA 5 dash to 3 dash UACGUA CGUAUG. Now next question number 26. Name the enzyme that catalyzes the transcription of hnRNA. And second B question, why does the hnRNA need to undergo changes? List the changes that hnRNA undergoes and where in the cell such changes takes place. These questions asked CBSE exam 2011. So first A answer is RNA polymerase second and B answer is hnRNA has non-functional introns in between the functional exon. To remove this it undergo changes. The changes that hnRNA undergoes include capping, that is methyl guanosine triphosphate is added to 5' end, tailing in which poly-A tail is added 3' end and splicing by which introns are removed and exons are joined. So now coming to the next question, number 27, states the difference between the structural genes in a transcription unit of prokaryotes and eukaryotes. You see here difference between prokaryotic and eukaryotic. This question is also asked CBSE 2014. The first difference of prokaryotics, this is polycistronic and eukaryotic is monocystronic. Second difference is number, no split genes present. The coding sequence is not interrupted. And second difference of eukaryotic is split genes are present. The coding sequence is interrupted to form exon and intron. Now, next question 28. A question is name the scientist who suggested that the genetic code should be made of a combination of three nucleotides. And second B question is explain the basis on which he arrived at this conclusion. The answer of A is George Gamow. And answer of B is he proposed that there are 4 bases and 20 amino acids, so there should be at least 20 different genetic codes for these 20 amino acids. The only possible combinations that would meet the requirement is combinations of 3 bases that will give 64 codons. Next question number 29. Explain the double function of AUG codon. Give the sequence of bases it is transcribed from and its anticodon. Answer is the double function of AUG codon. It codes for amino acid methionine. It is an initiation codon. The sequence of bases from which is transcribed is TAC. It is anticodon. Sorry, its anticodon is UAC. Now, next question number 30. Name the category of codons UGA belong to, mention another codon of the same category. Explain their role in protein synthesis. The answer is UGA. UGA is a stop or termination codon. UAA, UAG are other stop codons of the category. They prevent the elongation of the polypeptide chain by terminating translation. Now, next question number 31. Following are features of genetic codes. What does each one indicate? A stop codon, unambiguous codon, degenerate codon, universal codon. A stop codon does not code for any amino acid and terminates the synthesis of polypeptide chain. Unambiguous codon, one codon codes for one amino acid only. So when one codon codes only one amino acid, that is known as unambiguous codon. And now, what is degenerate codon? Some amino acid coded by more than one codon. So when amino acid coded by more than one codon, it is known as degenerate codon. And next, universal codon. Genetic code is same for all organisms. This is universal codon. All organisms on the earth, like bacteria to humans. Now, next question number 32. Mention the role of ribosomes in peptide bond formation. How does ATP facilitate it? This question asked CBSE exam 2010. So answer is there are two sites in the large subunit of ribosome for subsequent amino acids to bind to and thus be close enough to each other for the formation of a peptide bond. The ribosome also acts as a catalyst for the formation of peptide bond 23S RNA, 23S rRNA in bacteria is as ribozyme. This is act as enzyme called ribozyme. Amino acids become activated by binding with its tRNA in the presence of amino acid tRNA synthetase and ATP. Now, question 33 states the functions of the following in prokaryote. First, A tRNA and B rRNA. The answer of A is tRNA reads the genetic codes carries amino acids to the site of protein synthesis and acts as an adapter molecule. The answer of B is rRNA plays a structural and catalytic role during translation. Next question number 34. Draw the structure of tRNA charged with methionine. So you see here, this is a structure of tRNA and this is charged with methionine. Now, next question number 35. What is amino acylation? It states its significance. Amino acylation of tRNA or charging of tRNA is the activation of amino acids in the presence of ATP and their linkage to their cognate tRNA. If two such charged tRNAs are brought close enough, the formation of peptide bond between them would be favored energetically. Now, question number 36. Differentiate between codon and anticodon. So you see here table mention difference between codon and anticodon. The codon, the sequence of three nitrogen bases on mRNA that codes for a particular amino acid during translation is called codon. and you see anticodon, the sequence of three nitrogenous bases on tRNA that are complementary to codon on mRNA for a particular amino acid during translation is called anticodon. So anticodon is just complementary. So very simple. Now next question is 37. What would happen if histones were to be mutated and made rich in amino acid, aspartic acid, and glutamic acid in place of basic amino acids such as lysine and arginine? So answer is, if histone proteins were rich in acidic amino acids instead, instead of basic amino acid, then they may not have any role in DNA packaging in eukaryotes, as DNA is also negatively charged molecule. The packaging of DNA around the nucleosome would not happen. Consequently, the chromatin fiber would not be formed. So, what did you observe here? For the formation of nucleosome, it should be the two component of histone and DNA have a opposite charge, so they attract to each other. You know, DNA have negative charge, histone has positive charge. They attract each other to form a structure, and then nucleosome form uh, chromatin, and chromatin form chromosome. So this is basic need. Okay. Now question number 38. This question asked CBSE exam 2017. You see here, differentiate between the genetic codes given below. Question for A, unambiguous and universal. Question B, degenerate and initiator. So here you see the how to write by toppers. So this answer given by topper student. So you see how to write. So now next question 39. Name the enzyme responsible for the transcription of tRNA and the amino acid the initiator tRNA gets linked with. And B, question, explain the role of initiator tRNA in initiation of protein synthesis. This question asked CBSE Delhi 2012. The answer is, RNA polymerase third is responsible for transcription of tRNA, and the initiator tRNA gets linked with the amino acid methionine. The question of B answer is, the initiator tRNA, which is charged with amino acid methionine, reaches the smaller subunit of ribosome. Its anticodon UAC recognizes the codon AUG on mRNA and binds by forming complementary base pairs. The large subunit of ribosome joins the smaller subunit and initiates translation. Now, next question number 40. One of the silent feature of the genetic code is that it is nearly universal from bacteria to humans. Mention two exceptions to this rule. Why are some codons why are some codes said to be degenerate? So this question asked the CBSE exam 2014. So very good question. The answer is the genetic code is universal except in mitochondria and few protozoans. Very important. This genetic code is universal except in mitochondria and few protozoans. Some codes are said to be degenerates because some amino acids are coded by more than one code. Okay. So next question number 41. Given below is a schematic representation of lac operon. And here question A is identify I and P. You see here the diagram I and P. So I is a regulatory gene and P is promoter gene. Very simple. I is regulatory gene and P is the promoter gene. Now next question B is name the inducer. Name the inducer for this operon and explain its role. So answer is lactose is the inducer. Okay. Lactose is the inducer. It is the substrate for the enzyme beta galactosidase and it regulates switching on and off the operon. Okay. Now the next question is 42. How are the structural genes inactivated in lac operon in E. coli? Explain. This question asked CBC exam 2012. So answer is the regulator gene produces repressor which when free binds to the operator region of the operon and prevents RNA polymerase from transcribing the structural genes. Now, next question is 43. How are the structural genes activated in the lac operon in E. coli? Lactose acts as the inducer that binds with repressor protein that cannot bind to the operator and hence frees the operator gene. RNA polymerase freely moves over the structural genes, transcribing lac mRNA, which in turn produces the enzymes responsible for the digestion of lactose. Now, next question is 44. Genes contain the information that is required to express a particular trait. Explain. So, answer is the genes present in an organism show a particular trait by way of forming certain product. This is facilitated by process of transcription and translation according to central dogma of biology. So you see here, genes after transcription will form mRNA, and then translation to produce protein or enzyme. Protein trait product trait, an enzyme is bind with substrate and converted into product. Okay. Now the question number 45. A low level of expression of lac-operon occurs at all time. Can you explain the logic behind this phenomena? In the complete absence of expression of lac-operon, permease will not be synthesized, which is essential for transport of lactose from medium into the cell. And if lactose cannot be transported into the cell, then it cannot act as inducer. Hence, cannot relieve the lac-operon from its repressed state. Therefore, lac-operon is always expressed.